So, we've seen how the nucleus has a high degree of organization, where chromosomes form distinct territories in the nucleus, and active genes organize into transcription factories. What about other parts of the cell? What about cytoplasm? How does the cytoplasm look? Is it very busy, or is it mostly empty? Is it mostly water? And how do organelles organize? Are they just clustered anywhere in the cell? And these are questions that scientists also have. But as you zoom out more and more, it becomes more and more difficult to visualize how cells look, especially if you're trying to have a comprehensive view. For example, let's try and visualize the cytoplasm. You have to understand the size of the molecules, how many of a given molecule is in the cytoplasm, and any molecule that that first molecule interacts with. That's fine if you're only dealing with maybe 2 or 3 molecules, but there can be thousands and thousands of molecules in the cytoplasm. And so it becomes very difficult to bring all this information together, especially when the size of a molecule and maybe all the molecules it interacts with come from different papers. And so you have to, combining all this information is very difficult. But it is important. And so let's use an example, the insulin-glucose pathway. When we eat a sugary food, glucose gets deposited into our bloodstream, and we need the molecule insulin to allow glucose to be uptaken into our cells. So the mitochondria in our cells can use the glucose, turn it into ATP. And so what we're normally used to seeing is a very simplified diagram such as this. As this is the inside of a cell, and this is the cell membrane, and here we have this lighter purple is the bloodstream. And so what we're seeing is that inside the cell, we have a vesicle that has a glucose channel in its membrane. And so when the cell doesn't need glucose, it keeps these vesicles just in the cytoplasm. However, when insulin is in the bloodstream, it binds to a surface receptor. And when this surface receptor is activated, this vesicle is brought to the surface. The vesicle membrane fuses with the cellular membrane, and the glucose channels that were in the vesicle are now on the cell surface. And when they're on the cell surface, the glucose that's in the bloodstream is allowed to enter the cell and can be used now by the mitochondria and other proteins that need this energy source. So this is a very simplified version of this pathway. And let's look at a bit more complicated diagram. Over here on the right, there's a lot going on, but let's break it down. This right here is the cell membrane. We can see down here in green, we have one of these internalized vesicles that we saw previously that contain, here, let me get a pen, that contain these same glucose channels, just like we saw over here. Up here, we're seeing an insulin channel, an insulin receptor rather, just like here. And this yellow dot is insulin, just like over here. And so we can see that when insulin in the blood, and this is the bloodstream up here, that when insulin attaches to one of these receptors, the receptor interacts with many, many proteins, and the cytoskeletal elements are in charge of bringing this vesicle to the surface. The vesicle will fuse with the cell surface membrane. And once these glucose channels are at the cell surface, glucose, which is in the blood, can now enter the cells, go across the cell membrane, and enter the cell, and then go to the mitochondria or get used in the cell. And so this is a more comprehensive picture because you can see all of the thousands of molecules that might be affecting this pathway. And this is no small point. For example, if you are a scientist that is interested in curing diabetes, let's say type 1 diabetes, for example, characterized by resistance to insulin. So even though the body produces insulin and there's glucose in the blood, for some reason this pathway breaks down and glucose cannot enter the cell. And your hypothesis is that a protein has a mutation. And so which targets would you look at? Maybe the insulin receptor has a mutation, okay? And also the glucose channel might have a mutation. Very good hypotheses. However, this more complicated image shows that it's not just the insulin receptor and the glucose channel that are potential targets. There are thousands and thousands of molecules that are interacting with this pathway that could be potential targets for research. The large one is this skeletal elements that are in charge of grabbing hold of the vesicle and dragging it to the cell surface. And so these complicated and more accurate diagrams are very important, not just for communicating and showing how beautiful cells look, but for understanding how complex different diseases are and identifying new targets. If this is something that you're interested in, there's a master's in biomedical communication actually held at the UTM campus, and I've included a link here. And the diagram that we just saw was actually created by Dr. David Goodsell. He's a professor in the program. He does a lot of really amazing work. He's renowned for creating beautiful, beautiful images that help scientists understand and visualize the diseases that they're trying to treat. And so I've hopefully conveyed to you that making these images is very important, not just for communication, education, but for disease treatment. Now let's have a bit of fun. Dr. David Goodsell has made many, many pictures, and so let's have a look at some of my favorites from the gallery. They can all be found at this link right here. So first off is coronavirus, the reason why this is an online lecture. You can see here, this is the protein shell with genetic material contained inside and surface receptors. So this is in a blood plasma, and so this is a coronavirus surrounded by all the proteins in blood. Next we have E. coli. We can see that this is the cell membrane. These are cytoplasmic proteins, and this is in yellow, the genetic material. Bacteria don't have a nucleus, so there's no membrane separating the genetic material from the cytoplasm. And over here we're seeing the protein complex that forms the flagella. This is adipose tissue, and so what we're seeing here is a cell in the cell membrane, and here's a different cell with the cytoplasm. And so one way to storing fat in the body is in these large vesicles that contain fatty molecules like triglycerides. And so you can see that the body puts fat molecules inside these large vesicles. Over here we're having a look at cytoplasm, and it's not just molecules in the cytoplasm, but there's a lot of these cytoskeletal elements. And of course this varies from tissue to tissue and species to species, but you can see that there's a high degree of organization, not just molecules clustered everywhere, but cytoskeleton helps with cytoplasmal organization. Here we're seeing an immune synapse, so this is one cell down here, and this is another cell up here. These are cell surface proteins, and on contact they're interacting with each other and communicating. Here's another virus, a Zika virus. This is the protein shell in the bloodstream, again surrounded by blood proteins. And this is cell surface receptors interacting with the protein, with the virus rather, and it's trying to infect this cell down here. And here are two more synapses. There are many kinds of synapses, and here we're showing inhibitory and this should say excitatory synapse. You can see that both have vesicles filled with neurotransmitter, but in this excitatory synapse, what happens is the neurotransmitters are typically held within the cytoplasm, but then when you need it, those vesicles are brought to the surface and the contents of those vesicles are dumped into the synaptic cleft, and those molecules are now allowed to interact with the surface proteins on the postsynaptic cell, and the signal continues. And so this is how cells communicate. Again, I don't want you to be memorizing anything that I'm talking about here, but the main concept is that a protein, for example this one right here, it has a function, and so it has to be transcribed properly, but there are many factors that help that protein work. That protein has a function, but it's not going to work unless it localizes to the right place. And so this high degree of organization, cytoskeletal elements, other proteins, help bring this cell membrane, this receptor to its correct location, which is the cell surface. And so there's this really big concept, which is how proteins get to their function of interest. And that is protein localization, and that's our next topic.